When a virus infects a cell and synthesizes viral proteins, some of these proteins are degraded to peptide fragments. These peptide fragments are complexed with class I MHCs and displayed on the surface of the infected cell. Cytotoxic T cells interact with the virus-infected cells by recognizing both the viral antigen and the class I MHC. The cytotoxic T cell then releases cytotoxins which induce apoptosis in the infected cell and perforin which causes perforations in the cell membrane. Self-proteins are also degraded and the fragments presented on the surface via class I MHCs. This is not shown. Fragments of self-proteins are not recognized by cytotoxic T cells. Cytotoxic T cells remain intact, detach from the infected cell, and move on to other target cells.